Good morning, everyone. Sorry, we had a little public chat, but we are in the spirit of open science, so we don't have secrets. Yeah? Unfortunately, or maybe you think differently, today is the last day of the summer school, and we will end with two bangs. Yeah, nice talk by Martin this morning and Ilya this afternoon. Yeah, so thank you very much to all of you for being with us so long and steadily. Yeah. And maybe before I ask Martin to start, just a quick reminder, please this afternoon join us as well for the closing of the summer school. It will be a harmless, short procedure, but since we've invited a few stakeholders from the Department of Science and Innovation and the CHPC and so on, it would be nice to show them that that we were a big group when we started and we survived together until the bitter end. Yeah. So please, and I know that Benjamin shared already a few times the link, and I'm sure he will share it again later today. So please join us this afternoon. And it will be another link. It's not the link of Ilya's talk. It's a special link. So we'll have to go out of Zoom and in Zoom again at 3:30. Okay, Martin, this was my service communication of the day. I see you're already set for lecture 5. So please, Martin, over to you.
Okay. So this is the final lecture, and it's going to sort of be a bit different in character from the previous lectures, because I'm going to talk more about establishing phylogenies, using genomes in rather schematic terms. So it'll be a little like a computer science lecture with algorithms. And there are some sort of general things to take away, that some of these mathematical problems that we formulate using some idealized assumptions that are supposed to be solved by computer, they grow in a very nasty way with size. So as a consequence, one has to sometimes give up on finding the exact or best or optimal solution, and one has to sort of settle with a very good solution. There also is going to be some statistics involved, and I think at the end of the lecture you will get a flavor for the questions involved in establishing or computing phylogenies, but I'm not going to go through all the details in all the applications. So to just give a flavor of what the subject is about. So this is from a recent paper, and I chose this mainly for the image. And so here you have, this is sort of our family tree in terms of species, and here we are, and here's the chimpanzee and the gorilla, and these are the gibbons that mostly live in trees. And then this is a human-like fossil that is found in Italy, but there's no genome associated with it. So there are two trees that compete with each other. And this paper is by people whose specialty is the inner ear structure of the anatomy of the inner ear. So they make arguments pro and con versus these two trees. And a lot of, so what makes this different from the rest is this species is now extinct and there's no genome. So these sort of anatomical, more classical anthropology-based considerations must be used to sort of classify and establish a tree. But with having genomes, there is just a wealth of information about differences that can be exploited, and classification and ancestry can be established with a lot more confidence. So this is sort of one application. And there has been a sort of shift from before all this data has been available from molecular biology. Classification was, very subjective, and there wasn't really much to go by. And now a lot of these problems can be translated into mathematical or computer science problems, and one can make these trees and establish what species to group together, based more on how they evolved, the history. Because even with just having the genomes today, well, we could guess a bit what the past genomes are, but we can sort of establish a tree with a sort of moderate or high degree of confidence, depending on the situation. So this is sort of molecular paleontology. This is the application here. So what we're not going to talk about is really how to do this in detail. So if we look at species that are very far apart from each other, the size of the genome is going to be very different. So, you can find, if you look at vertebrates, certain proteins or certain sections that are sort of conserved, that they're necessary. So hemoglobin is something that every organism that requires blood is going to have. So you can sort of narrow in on these certain regions for species that are, very far apart, and try to establish trees. If we compare species that are very far apart, we sort of find that the sort of non-coding part of the genome, which may or may not be noise, but is less important, that is going to vary, and that can be cut out and changed in length without too much problem. Whereas the sort of genes that don't provide characteristics that are sort of add-on options, but if you didn't have them, you would die, then those are going to be conserved. And looking at these comparisons, it's not just sort of making these cute trees and, this is obviously very interesting, but it also sort of teaches us about our genome if we're interested in medicine and medical applications. Well, which genes are very important, and more so what part of the gene is the most important. So if, certain proteins in the coding never change, then that's sort of suggesting that, oh, well, if that changes, maybe the protein will fold in a completely different way and the gene won't perform its function. Whereas if you see a lot of variation, then you might suspect, well, this isn't that important. So that's sort of a reason why there's a great interest in sequencing a wide variety of organisms and comparing genomes, rather than just focusing on our own genome and saying, well, we're interested mainly in medicine, so let's look at our own. So if you look at these differences, you see what's important. So the models we're going to use to talk about algorithms are going to assume that the genome size is not important and changes occur randomly at any place. And that this is just to sort of simplify the problem, create a model problem so we can look at the algorithms, and we can look at also the methodology. So there isn't really an agreement on what is the best way of saying which tree is better than another tree. So this is sort of one type of application where we have genomes today. There also is sort of archaeological DNA where, from the Neanderthal, if a body has been frozen, the DNA can be taken out and sequenced, or at least in part. And there's also some interesting work about the plague, for example. So some DNA from the plague was taken out of a tooth and compared to that. This is, from the 1300s, and compared to the current genomes to establish, well, what was the origin of the Black Death. But in general, a lot of this information will be not available. Okay, so let's look at a different application. So this is a sort of the choice is a lot less random. So this is a very nice and important article from the group of Tullio de Oliveira at UKZN at the medical school. And this paper was submitted to Nature, even though the publication date is now, it was submitted in October, but I think there's also some December data, maybe with the revisions. It's not super clear. But here you can see that there's sort of a partial tree from the COVID-19 virus, and the tree starts the ancestor in around December, or in so this would be the Wuhan virus. And what is sort of unusual about COVID-19, at least from what we sort of understand, is that it came from one place. So the hypothesis is that there was sort of a single jump from, the bats to humans, and, there might be some intermediary animals, but this is sort of jump is something that doesn't happen all the time. So it's sort of a big deal event. And all of the new virus can trace its origin to this one event. So this means that we actually know the root of the tree. So that's something you can download from the internet. So I'm sort of contrasting this to the previous slide. And there's also data all along in time. So when we look at these the molecular paleontology type examples, we sort of know the boundary conditions today, but we don't have all of this genomic information in the middle. So there's a huge amount of information, and the changes here are very small. So this is a problem that is quite different in character from the previous problem that we were discussing. And the methods have to be different. So let's just go over what is being shown in this diagram, and the data is rather old, so one could come up with better plots that are more recent, but this was sort of the first observation. So here is sort of a selection of a few thousand little circles, which I don't know exactly how they were chosen. There are a few 100,000 genomes now available that you can download. So this isn't everything. So all of these gray dots are genomes that came from outside of South Africa. And then the red is in South Africa. I'm sorry, the blue is what is seen in South Africa. And then the red are the new variants that have appeared in South Africa. And what you can see, and this might not be completely accurate, is that some of these strains have died out, but then you see the red sort of taking over. And this will, of course, as we've all heard in the news, there are strains that have a higher R than other strains. So there's sort of this competition in the UK strains, and some of these new South African strains are sort of taking over, and Brazil's not left out, and there are new things going on. So the sort of lesson from this is that there are useful things that can be found out by sequencing and studying all of the COVID-19 genomes. And before these mutations were seen, I think some people thought that this kind of research was not so important, and is it really worth the money? And at the beginning, these changes did not have any observable consequences, so they were perhaps useful markers, and there are things that can be done with these markers. So the same group wrote a paper looking even earlier, looking at how many times was COVID-19 introduced into South Africa. So that provided interesting information about, how important it is to close borders, and is that valuable, etc. But in those cases, there was no observable evidence that the different strains coming from either directly from Asia or through Europe or the United States, that they had different properties, both with respect to their contagiousness or their virulence. So this slide is to convince you that this is a useful exercise, and just like the previous one. And also the other message is that there isn't one phylogenetic problem, and it's something where you can just you take your sequences, you put them into the software, and you just get your answer. The information available depends on the problem and what is the best algorithm. Okay, so now we're going to go a bit into the algorithms. So this is going to be, rather simplified, because there's no sense in drawing huge trees. If we can sort of define a problem with a small tree, let's try to understand that first. So let's just take here 3 species, and let's do the first problem where you sort of know the genomes today, and we'll just denote them abstractly by A, B, and C without saying what information there is. And we're also going to assume that, well, a few things, that there's a common origin. So these are going to be rooted trees, and then they're binary trees. So there's, at each branch, there's just 2 offspring, you might say. So time flows forward from the root upward. And what we see in this transparency is, in this case, there are 3 trees that compete with each other. So this would be a very simple problem, in that all of the problem would be, well, what is the right metric to use for, comparing these 3 histories? And that is a sort of complicated problem if the species are quite distant, so their genomes are different size and all of that. So that's sort of one sort of problem. But what I want to focus on now is just the trees. And maybe where I'm going to go right now is more the counting the number of trees, and we can see that this problem is not going to be something that we can solve in just having a looping over all possible trees, having some figure of merit, and then what is the best. Or you could do a sort of Bayesian approach and or probabilistic approach and assign probabilities to the trees. Because if you think about it, if mutations are random events that occur and not by design in general, then it's not a foregone conclusion that if you have the information, there will be a correct answer. So there should be room for saying, oh, well, we believe this story with a certain confidence, but then there could also be another story. So we're going to explore if we have N species where there's a very large number, how many trees do we have to consider? So this is a computer science problem, and we'll see that this is related to the traveling salesman problem, or if we wanted to write a computer chess program that, for example, would beat a human. Well, part of the reason that has been possible is because computers have gotten faster. So computers are smarter than people in chess in some cases now, but also not looking at every possible continuation of the game. So you could think of a chess game as sort of a tree where each successive move creates branches, and then the tree just grows. And what you're trying to do, if you wanted to get the perfect problem, the perfect solution with 100% confidence, is sum over all possible games. It's not even clear that that process would converge, that the number of trees isn't infinite. And that's just not practical. So you sort of need to prune the trees and eliminate stupid moves as early as possible and not explore those branches. So this is quite different from most numerical algorithms where you just get the answer, and there are various tricks. Okay. Yeah, I think this has the points that I just said. And if you want to learn more about this subject, Joseph Felsenstein is a professor at the University of Washington, and who's now retired. And this book, Inferring Phylogenesis, it's rather theoretical, and doesn't talk about the applications, but it's rather simple. It's written so biologists can understand it. And historically, he was one of the first people to come up with algorithms that would run on computers in a time that sort of converged and that people could use. And this, at the time, the data wasn't available, and computers were in their infancy, so there wasn't so much interest. But then this is sort of his papers are some of the most cited papers. So this is a good place to learn the sort of theoretical, mathematical aspects of the subject in a way that is accessible. Okay, so we're just going to, for simplicity, look at binary trees, meaning that you only have 2 branches as you go forward, and that there's a root. And we're also going to assume with some of the methods that there's no information about the intermediate genomes and the intermediate times of these branches aren't known. So you might know from paleontology when these different divergences occurred. So this is a way of checking some of these molecular results, but we have to sort of choose a simple problem to illustrate some of the methods and tricks that can be brought to bear on this problem. Okay, so let's establish the rules of the game. And these are not going to be the rules of the real game, but this is sort of what applied mathematicians would call a model problem. So you can understand and explore algorithms and the variations to take into account more realism are somewhat straightforward. So we will assume that the genomes are made of sequences all the same length of the 4 letters A, C, G, and T. And we're going to assume that all the transitions to are equally likely. So that might be a bad assumption. We sort of know that some of the changes are going to be lethal, but if they are lethal, then they won't be observed. So this might not be that bad of an assumption. There's no taking into account of change of lengths, that there might be some correlations in the mutations, that there are sites that are more likely than others, etc. So these assumptions have to be made with a certain amount of trepidation. And so we need a stochastic rule in order to have a well-defined problem. There's also this phenomenon called homoplasty, which means that some changes can occur independently without a common ancestor. So for example, if there is sort of selective pressure for an organism to change and adapt to acquire a certain feature, the same thing could happen independently as a result of the selection pressure rather than common ancestry. So that's one of the possible dangers, and some similarities might be given too much weight. So one very simple way of avoiding that is to sort of divide the data and see whether you come up with the same trees if you ignore or take into account certain genes and that kind of thing. So that's something that can be explored and in principle also detected. Okay, so let's talk about the first class of methods. And as I found out from an oral history interview with Joseph Felsenstein, there were tremendous fights about, what is the correct method and the basis, maybe more so in this field than in other fields. But this is a principle that, find the history that requires the least number of changes. And if you think about it, that's not necessarily going to give you the right history, because if these changes are random, well, your most simple history might be too simple, and there's no reason to believe that the true stochastic process had to conspire to produce the simplest story. So there has been a lot of work on justifying this assumption, and there's some examples where even in a sort of large limit, it can give you the wrong result. So if we look at statistics, and this is more frequentist statistics, there is this quote, principle of maximum likelihood, and it doesn't, in the general case, give you the right answer, but then you can show that in certain cases that asymptotically, when the amount of data is large, it'll be very close to other optimal answers, and it's sort of easy to search for. So that's sort of what you might hope for. So here we write what A, B, and C are, and I'm just going to simplify with there's zeros and ones rather than 4 bases. And I sort of worked this out, and this tree gets a score of 2, and that I can go from a root genome to these 3 with just making 2 changes. Although the interesting thing in this example is that I cannot reconstruct the root genome uniquely for this score. So this is going to sort of be a general feature, and a requirement to find out, what is the evaluation of the tree doesn't necessarily mean filling this in. So we have in our sort of ideal deterministic algorithm, we have a loop over all trees. We give them a score, and then there might be a tie for what is the best tree if the score is the same, or there might be a unique answer. But there's no probability involved, because there's no notion of stochasticity in here. This is our sort of ad hoc, seemingly reasonable principle, but it cannot be justified in terms of a plausible stochastic underlying model. Although in this field, especially in the beginning, the emphasis was on, getting results that could run on computers at the time, and, not necessarily getting the perfect answer. So this is, in some ways, the simplest method. And I'm going to explore this here almost exclusively just to show you some of the algorithms. Okay, let's talk about sort of a subproblem. So here I'm going to just look at one piece of the genome and you know, one bit. So we're going to have the 0 and 1. And if I have a complicated tree, so with the binary trees, they're basically with each boundary endpoint or species, I'm adding an interior node. So the number of choices for, you know, what these genomes would be just becomes huge as the size of the tree expands. So you would like to find a way that you don't have to, in your program, put a loop over all these 2n choices and then do that for each bit in the genome, and you would get a program that would be completely unmanageable. So there's a simpler method, and it's sort of a style of programming called dynamic programming, which was introduced in the 1950s by the mathematician Richard Bellman. And you'll see that this exponential problem gets reduced to a problem that is linear in n, which is a nice thing. So the trick is to break the problem into a sort of nested series of simpler problems. And this seems sort of too good to be true, almost. But the sort of trick is that, well, we don't need to know what the intermediate states were or could have been. We just want to score. We want to know what is the number of changes. So dynamic programming, one way to think about it is that it's divide and conquer, or it's somewhat recursive. So we want to break this problem up. So here I have these two boxes, and the score flows downward, and the left box is, well, if I just look at the transition from, this binary choice here, what would the score be if the bit here were 0 in the left and 1 in the right? So I add to the sort of score how much have you lost. So once you're on top, you haven't lost anything. So here, I could make this either a 0 or a 1. And it's sort of equal. It's a lose-lose game. And here it's a lose-lose game. And then you can think of this as that we're pruning the tree, and instead of having this where the penalty would be 0 and infinity, or infinity and 0, we, rather than put a specific result, we have sort of two boxes like this. So we've gotten rid of this branch, we've gotten rid of these two branches, and our new tree is this, this, and this. So then we find, well, if we set this equal to 0, we have to have a transition here, and then we have to add the penalty from that already exists from 0 here, so it's 2. Whereas if we would set this equal to a 1, there would be no penalty here, and you just the existing penalty, so you carry it down. So then we've pruned off this entire thing, and then we do the same thing if we have 0 here based on these boxes, we get the score, and in the end we know what the best choice would be at the root of the tree, but we've sort of lost the information, or we don't really care about it. So this tree gets a score of 2. And here is the sort of basic scheme, that you're removing nodes and you replace the nodes with these penalty functions, which are sort of cumulative, adding the existing penalties. So there are 4 choices to examine, and then 2 choices here. So there are 8. Every time you do this operation, you have to look at 8 possibilities, and we can just write this in terms of 2 functions. So we've taken an algorithm where if you would do it in the simplest way, it would be exponential time, and the complexity, which we will quantify as the number of species in the tree, and here it's only linear. So that's pretty successful. In this basic dynamic programming, it's also used for aligning genomes, so it's deterministic. So if you want to find out the insertions and deletions, well, there would be a huge combinatorial cost. So if you wanted to align 1,000 with 1,000, you couldn't do it by brute force. But this dynamic programming, where you're trying to embed, well, here we generalize by not having definite values at the endpoints, but we're sort of embedding problems in other problems, and it's sort of like a Russian doll thing. So this is a paradigm for finding clever algorithms to do things that would be hard to do just using brute force. Okay, so we have solved somewhat adequately assigning a score to a tree. So there's no longer sort of exponential time required. And I mean, the rule of thumb is that if something doesn't scale as a, polynomial, or even more stringently, a polynomial of low order, you cannot do. You cannot do the problem in a 100% reliable way. So now we're going to look at how the number of trees grows. So we want to look at the slope over trees, and we want to see, as the number of species increases, what is the the function is number of trees of n. So we stick to rooted trees, and there's this binary branching. And if we have n is equal to 2, then there's a unique tree. So there isn't really much to talk about. Here's the tree on the first slide, and you have to think a little about what is the same tree. So if I write this as B, A, C, well, this permutation doesn't really do anything because A and B have a common ancestor. So you have to think how you want to count. Here we need to swap B and C even to draw this tree. So what is a tree for our purposes is not necessarily the way you draw it on the page. So you can just do this by sort of simple combinatorics, and you choose one pair, and then the third one is completely determined. So this would be 3 times 2, but we don't care about the order, so we divide by 2. So this is pretty easy to get correctly. This is a problem that Cayley worked on and others, and it was sort of more recreational mathematics, and I think around 1857 or something. And he wrote some papers with generating functionals, but there's a simple way for this particular counting problem. Okay, when I did n is equal to 4 and 5, I got the wrong results a number of times, so it becomes rather tedious. So here we can see that if we have this basic pattern, where there's a pair and then it gets paired with something else, which gets paired with something else. If you think about it, this is the, all of these permutations are meaningful, except A and B can be switched with this initial pairing. So we take 4 times 3, oops, there should be a dot here, times 2 times 1. And divide by, I didn't write it out, but divide by 2, so we get 12. So there are 11 other permutations of this. Then we could pair in 2 groups. So we can assign 4 factorial different values of A, B, and C, and D to this top line. But pairs here, the order doesn't matter. So we get a factor of 1/2 here, and we get a factor of 1/2 here. But then we also don't care about how this is paired. So we divide by 8 and we get 3. So there are 15 different types of trees, and I won't. This is the same thing for 5. So this is the kind of problem where you do a few examples and then you try to come up with a trick. And let's see, I did this one. Yeah, here you get an 8, and there's, because this is the same sort of sub-problem, and then there's something left over. Here you divide by 4. So I get the sequence of, if I do the counting, and then it's times 3, times 5, times 7. So you sort of see a pattern here, and you would like to come up with a simpler argument, because it seems that each n has a more complicated shape, and you need to get the combinatorial factors right. Okay, so this is what we sort of found by trial and error. And here is a sort of another proof. So let's, well, first let's count the number of edges. So in a tree, and you can see that you're adding, if I add another point, I bisect an existing edge and add another edge. So you're adding 2. So you get this this number of edges. And then what we're going to do is we're going to say, well, let's say this is A, B, C, and D. And then we're going to add another species. So you could connect it here, you could connect it here, you could connect it here. Each edge can be bisected. So then this gives you the well, it would be for one less, how much the factor by which you've increased the number of trees. So this is sort of like the factorial, and it grows very quickly. So I guess what I'm showing you is that with these trees, you cannot enumerate over all of the trees, that you may as well give up doing an algorithm in this way. So, we can estimate this as a sort of a logarithm integral that is off by a constant factor, and you get something like the Stirling approximation without the prefactor and without the different asymptotic corrections. Okay. So we need to find an algorithm that is either going to be clever and somehow identify that, there are a lot of trees that you don't have to consider, and that's, a very logical possibility. So I haven't proven from a theoretical computer science standpoint by any means that you cannot make a more efficient algorithm by sort of being clever and coming up with some reason why evaluating a subset of the trees is sufficient. But I've sort of suggested that the brute force method does not work. It doesn't scale in a good way. So let me digress to a slightly different problem with which has a sort of same characteristic. So we'll look at the Hamiltonian path problem, which is something that was first written about by William Rowan Hamilton, who was, the father of the Hamiltonian formalism and quaternions and all sorts of stuff, but he first looked at this problem. So we throw down points at random in a plane, or more generally, we could just come up with some metric. What is the distance between them? And you need to find the shortest path. So there are 7 factorial such paths. And, with traveling salesman, the path goes back to the beginning. So this is sort of another sort of famous computer science problem. But here we just look at a path that starts somewhere and then ends somewhere and find the shortest path that visits each point exactly once. So we throw down points at random and we explore all paths that satisfy these conditions. And we could do n factorial program. It would run, it would be easy to program, but it would run very slowly at a certain n, and you increase the computer power by a lot, and you can't increase n by a whole lot. So can we do better without coming up with something that is sort of a bit ad hoc and comes up with a good enough solution, but not necessarily the best? I just put this, I don't know the answer to this question, but I wonder if there are any quantum algorithms that could do this with quantum parallelism. So my argument about why you can't enumerate this is assumes a sort of Turing machine computer. So let's represent all the possible paths as a tree. So I'm going to show you a sort of shortcut where you can save a lot of work, but not necessarily enough to really change the game. But this would still be deterministic. So we start out by choosing the starting point. And so here I use the, traveling salesman that he has visiting cities, and he has a plane that flies, as a crow flies distance between the cities, etc. But you can represent this n factorial as a non-binary tree where there are n branches to n nodes, and then each of these, let's call them level 1 nodes, there are n-1 branches leading to nodes that in turn have n-2 branches, and then at the end, you only have 2 cities to choose from. So you have a sort of distance from each path. So we can ask, well, do we really need to explore all of these paths? And the answer is no. That if we have start exploring, we're going to have sort of a running solution. Well, what is the shortest path? So basically, you traverse a tree, when you're traversing another branch that hasn't been explored in your algorithm, if before you reach the top, you've already exceeded the distance of the shortest path, you can just stop there and you can prune off all of the branches, and you can also imagine that this is carried out in parallel, so that there are several workers, or whatever you want to call them, traversing different paths, and then they communicate, well, what is the shortest, but they just prune and backtrack when what they're exploring exceeds the shortest length. So this is sort of not a completely well-defined problem, because the exact distances between the points are going to determine how successful this strategy is. But the empirical evidence is that, it's good, but it doesn't work that well. On the average, the difficulty climbs too fast with n, and you're in trouble. Okay, yeah, here I drew out for n is equal to 4 all of the paths, and you can sort of look at this and sort of think, well, what would you could follow this, then you go back, and this might be longer, but you're basically pruning, then you go this back here. Well, if this is longer than your record, then you can just cut this out in the same here, but you might have to go all the way up to the end. And then, if this is shorter, you can prune your best path here. So you're basically traversing and pruning your tree until you get the optimal solution, or a number of solutions that are equal. So you would end up with a tree that could be, unary at some parts after all the pruning, that would have all the, either just the optimal path in terms, which would be a sort of unary tree, or there could be several branches, or it could have different structures. Okay, I'm just talking about this problem in order to, what we're going to want to do is we're going to want to make a tree that creates trees, or enumerates all the trees, and then sort of improve on that. So if we were doing the traveling salesman problem, we here there are points on a plane, and we could, for example, always go to the nearest point and start at different points. So we would that way be starting with solutions that are not sort of ridiculous zagging back and forth, which would be the most solutions. Most of the possibilities, and then explore minor rearrangements. So this is a little like what you would have in simulated annealing with a continuous problem where you want to find the maximum of a function, but there could be many local maxima. So you sort of climb up to the top, but then you also have an algorithm where you might not always go uphill, but go downhill with a certain probability. And, by reducing that probability, you can hope to converge toward the right solution. And in the Felsenstein book, there are a number of sort of schemes for finding nearby trees that to explore that have this property, and you can sort of simulate with trees that have been generated stochastically and see how well you do. Okay, let's go to another problem, and this would be for COVID. So the problem is a bit different because we have the intermediate genomes. So we can start connecting things without having the entire tree. And then the other simplification is that there are not very many mutations and the genome is very long. So you can assume that at least at the beginning of the epidemic that there aren't any cases or very few cases where a change has occurred twice. And the root genome. So here we've sort of enumerated things that are different from this sort of paleontology, reconstructing natural history problem where we just have the, data at the final time and we're trying to reconstruct things. So we can, under these assumptions, for example, just look at the set of mutations and call this M sub I. So this would be the, number on the sequence where there has been a mutation. And since there's diluteness, this is not going to happen twice. And then we can, if the set of mutations a basis relation, then you can sort of tell that the ancestor was the smaller set. So you have mutations that just add on. And if you have enough data, you can sort of order the mutations so that in one of these sets, we can determine, well, this was the first mutation, this was the second, et cetera, et cetera. So these become completely ordered. Then if we have, under this dilute approximation, two sets of mutations where the intersection is non-zero, but there isn't total inclusion, then we can conclude that the intersection of these two sets must have been a common ancestor. Under the above hypothesis. So this is more a deterministic algorithm, but when the hypotheses are violated, when there have been too many mutations, then this will not always be true, but become sort of fuzzy. So let me just construct, take a sort of simple example. So let these letters be observed mutations, and I'm just going to sort of choose them sequentially. So we know when there's a single mutation with respect to the root genome, that this is going to be, directly connected to the root. So we can fill these two in. Then when there's, with the AB and the A, with the inclusion, we can tell that the B occurred first. Now, if in the unlikely event where B is also present alone, which is very unlikely, then this would have had to have occurred. That this can occur in more than one branch, but that's sort of unlikely. Then we also have the D and the E, but so we know that these sort of longer sequences come off of this, but we know that the E is later because of this inclusion. Here we have to come up with, we have FG and FGH. So we know H came later, but we can't distinguish between these. Did F come before G or G before F? So that's just from the insufficiency of the data. So we can, we're going to have to put these two mutations as something, as far as we know, they happened simultaneously, but we can't put them in an order. And then we can have a lexicographic order for this dictionary. So, A is before C, before FG, but then you add, so this, all the things that come after. So this tree can be uniquely represented in terms of such a dictionary. And then you can sort of see that you can insert additional data into this dictionary structure rather efficiently. So this algorithm works when there are not a lot of mutations and not a lot of, also that there's not a lot of data. Okay, so in terms of analysis, well, there isn't complete coverage. And also, if you look at the data, there's a lot of bias in that people investigate clusters. So you tend to have a selection effect toward genomes that are related. The errors in sequencing and the data is incomplete. So there are gaps, so you can't always tell, well, is a certain mutation present or not. So you can sequence COVID-19 for less than, well, let's see, 100 euros. So I guess that would be about 2,000 Rand a genome. So this is sort of the wholesale price. And so with today's technology, it's not that expensive. Let's talk a little about, well, some of the theory, and this depends a lot on the specifics of the COVID-19 virus. So there are two time scales. What is the time scale between observed mutations, which is about 3 weeks, and what is the time scale between spreading, going to another person, which is about a week. So that means that a lot of transmissions will not involve a change in the genome, and every once in a while a new marker will have been put on. So we have in our mind this sort of stochastic model, and that seemed to have been consistent with the data for a while before all this new strains came along. So the hypothesis would be, well, the mutations that we see do not result in any great change in fitness. So a change in fitness would mean that you would find one part of the tree expand at a much faster rate than another part of the tree. So that's what you would see or look for if you were trying to make a sort of data analysis method that looked for new strains. Okay, so how could natural selection come into play? One important thing is that when people spread the virus, what is spread in a droplet or whatever is, not the whole biodiversity in the infected person. So if you have, millions of viruses in your body, if you're infected, within your body, there's going to be a huge amount of diversity. So there's sort of this tree developing in yourself, and there can be, and is going to be selection. But then you're just drawing a small number, maybe even one viron that succeeds in infecting somebody else. So all this natural selection is a big failure unless it's managed to, within the time you're contagious, become sort of majority or have large representation within in your body. But if you have somebody who is infected for a long time and neither dies nor gets well, so the hypothesis might be someone who's has a impaired immune system, so that they never really recover, but for some reason they don't have that many symptoms, then you have much longer time for this exponential selection to take place. And this can explain, this is one of the explanations for how these new strains would come about. Another explanation is that, for example, if people are given convalescent plasma, which is sort of similar to, mathematically to antibiotics, well, there's sort of the selection, and what is resistant against the body's defense will remain and be selected. So it is possible that treatment can, some of these treatments can favor natural selection or deselect things that are can be treated with drugs. And there's some studies where people have been given this plasma and then they sort of do a lot of sequencing and they see that the strains within an infected person change. So these are interesting questions. So we've sort of gone to something where the COVID-19 genomics were just sort of markers with a sort of neutral theory of molecular evolution, to where the fitness considerations are the most interesting thing that is, and significant thing that is happening now. Okay, so this is has been a very bare bones introduction to phylogeny, and we only, mainly for lack of time, explored the parsimony methods, where we're looking for the simplest story. But there are other methods like likelihood that apply a probability to different transformations and try to come out with probabilities for different histories. So this is sort of a method of principle, and obviously that makes more sense than parsimony, which, if you think about it, it is not a reasonable exact principle, but it could asymptotically be a good choice, especially if they're algorithms. Then there are also distance-based methods where you just make a table of how far apart different genomes are to each other. Okay, we didn't explore realistic models of genetic variation, and that's sort of a big issue here if you want to do anything practical. We saw that this is the kind of problem that gets hard very quickly. Someone has to have some sort of clever sampling. And we also saw how dynamic programming can be used to come up with faster algorithms without any compromise in the quality of the results. So this is sort of a nested Russian doll approach. Okay, so what did we not discuss? Well, these are the two other classes of methods. Well, we talked about genetic clocks, and this can be used, for example, if we would take these primate histories and we had derived them in terms of parsimony, we could see, well, is the genetic distance between the different organisms at the top of the tree, is it consistent with the inferred tree? So this allows for some cross checks. Another thing we didn't deal with is how to deal with diploid population. So we saw that there's, you know, if we have a well-mixed population, then there are these different alleles that are sort of floating around, and they occur, you know, with certain probability. So there are also methods where rather than taking one genome from each group or species, one just looks at the probability distribution of alleles, and you can study, for example, human migrations in much the same way, where the allele frequencies takes the place of the genome. So that's another that's another facet of this area. So this concludes my 5 lectures. I hope that I've conveyed that there are a lot of different facets to this, that it's not all molecular biology, it's not all computer science, it's really or statistics. It's a multidisciplinary field, and there are a lot of new questions that are becoming feasible because there is so much genomic data of all sorts. So this is an interesting area to look at, and this is not something where you know, all the problems would have been solved 100 years ago because the data hasn't been available. That said, it's very interesting that a lot of the theoretical work seems to, and ideas seem to have preceded the data, and there's now more interest in this field because applications are useful applications are becoming possible just because of the low cost of sequencing that sort of changes with Moore's law. So this might be the time to come up with impractical applications that people will say are useless today, and then in 10 years, everybody will be doing what seemed impossible. So thank you for your attention, and I would like to thank the organizers for giving me this opportunity to present these lectures to you. Thank you.
Yeah, Martin, no, thank you very much. We have to thank you for preparing this wonderful set of lectures. It was really interesting, and we all learned a lot. And you know, it's always amazing to see what theoretical physicists are able to do. Yeah, so there is nothing that can stop us. So thank you very much, Martin, for that. I'm sure everybody enjoyed these lectures as much as I did. I don't see at the moment many raised hands or dozens of questions in the chat. So maybe let's give a minute to the participants to collect their thoughts and use the opportunity this last opportunity for the school to ask you a few a few questions. No, Martin, it was really, really very nice. And I think you should write a book to address even a wider audience on this topic. Oh, I see. There is a first question in the chat by Tebogo. Let me Tebogo, since it's the last lecture, let me give Tebogo the right to talk. Tebogo, you're welcome to ask a question yourself. You just need to unmute yourself. Okay, if it doesn't work, the question is, are there any chances of genes being transferred horizontally? Won't these affect phylogenetic trees?
Okay, this is all the questions have been excellent, and this is an excellent question. So
we have a candidate for the book voucher.
So the point that is being made, and I can sort of give an example where with viruses that so the sort of naive theory of how genetic information transfers is that it's this tree, and it just comes from, you know, random mutations or, you know, inheritance from ancestors. So if a certain segment of DNA is copied to another genome, so horizontally means across the tree, not respecting the hierarchy, then this is going to, you know, completely mess up the analysis. So this is the point that Tebogo is making. So one way this could happen is that if, and does happen, is that part of the, say, human DNA, I'll just use people as an example, could get incorporated into a retrovirus, which in turn infects somebody, and then when the retrovirus genome gets incorporated into somebody else, that segment is so-called transferred horizontally. So that breaks the rules or the mathematical assumptions in perhaps the most serious way. Or we don't even have to we could have genes that just come from the retrovirus. And I don't know the numbers offhand, but if those get incorporated into the germline, then you know, there's a unobserved retrovirus that is in the tree. So won't these affect the phylogenetic trees? They will. So the way to, the way to sort of guard against this is I think if we were doing human phylogeny, and there are, different problems with that, we would sort of subdivide and see, well, are there certain, we say do tree for each chromosome, and is it the same tree? And if there is a sort of big difference, then one might sort of look and at well, where is that coming from? And that would be a way of finding horizontal transfer. So suppose there were genes or parts of the DNA that gave you a completely different tree from the rest of the genes, then that would be evidence of horizontal transfer. So in principle, by having lots of data, one can sort of figure out that this is going on, and you can get interesting additional information. So it's also an opportunity to discover horizontal transfer. And let me just mention that, when we talked about bacteria and exchanging plasmids, which are extra chromosomal DNA, that's one example of horizontal transfer violating the tree, and that's considered very important.
Okay, wonderful. Martin, I think I'm just worried about the time. Unfortunately, all good things come to an end at some stage. So even this series of lectures. So Martin, thank you very much again for the really interesting and exciting lecture series. And I hope we will see you later this afternoon for the closing of the school at 3:30. So thank you very much.
I'll be there. And thanks for the invitation.
Thank you very much. And thank you to all the participants, and thank you, Tebogo, for the nice question. Yeah, thank you very much. So have a good rest of the morning, and we will see you again relaxed and receptive again at 2 o'clock this afternoon. Thank you. Have a good morning. Yeah. Bye-bye. Thank you, Martin.